who want to follow the paper. And it will be presented by Jain Mung from University of Southern California and is entitled A Non-disruptive Technology for Robust 3D Tool Tracking and Ultrasound-Guided Interventions.
Hi, good afternoon. So if I could direct your attention to the screen, what we have here is a 3D ultrasound movie of a pig's beating heart. You can see clearly the chamber of the heart, the ventricle. Down here is the myocardium. But what's not so obvious is the fact that there is a catheter inside this pig's heart. And that catheter is right here. So you see it now, and there it goes. So you can imagine this could be a serious problem when it comes to these cardiac interventions guided by ultrasound. So our work addresses this problem. Again, what you're seeing here is 3D ultrasound, 3D ultrasound movie. But instead of that 3D rendering that I just showed you, what you're seeing now are two orthogonal slices through the volume in azimuth and elevation. And you can see clearly the chamber again and the heart walls. But what's different about this view is now you can see unambiguously the tip of the catheter as it moves in and out of the chamber. So that's pretty much my work in a span of about 90 seconds. But because I have another 10 minutes or so with you, I'll give you the rest of the story. And the rest of the story is titled A Non-disruptive Technology for Robust 3D Tool Tracking for Ultrasound-Guided Interventions. My name is Jain. I'm a graduate student at the University of Southern California, and I'm presenting this work on behalf of my former mentors at Philips Research, Francois, Vignon, and Ameet, Jain. It's my pleasure to be here with you today. So the rest of this talk will follow more or less traditional structure, and I'll move on by giving you some background in terms of the clinical significance and the current landscape. So 3D ultrasound is positioned to become the standard of care for particular cardiac interventions, specifically transesophageal echo, where the heart is imaged with an ultrasound probe that's placed inside the esophagus, has demonstrated benefit with structural aortic, mitral and aortic valve repair. In 2008, Balzer et al. Detailed their experiences using this 3D ultrasound in a number of different cardiac interventions. But the problem is still there. Even if you have this nice 3D ultrasound visualization, the catheter tip is hard to see. And really, this problem is fundamental to ultrasound. Ultrasound was designed to image soft tissues, and these interventional tools are not soft. They're either metal or plastic. And in general, this is, there's a clinical need to simultaneously visualize tools in anatomy. And I think this whole community here is familiar with some of the modalities that we have available to address this issue. There's the traditional fluoroscopy. There are EM trackers that you can combine with image fusion techniques. There's a new effort to use shape sensing fibers to give you information on the shape of a catheter. There are also ultrasound-only based approaches, such as beam angling, where the ultrasound imager will angle its beams to provide a more favorable imaging parameter. There are also these so-called echogenic surfaces where tools are specially textured to show up brightly on ultrasound images. But our approach is to use these small sensors that we place on these interventional tools. And various attempts at this sort of approach have been made over the last 25 years or so. But with our work, we hope to show that we're the first to demonstrate real-time, robust, 3D accurate tool tracking using ultrasound only. So next I'll detail some of the technical aspects of the system. Ultrasound imaging probes transmit precisely timed and focused acoustic beams that raster across the ultrasound field of view in range, elevation, and azimuth. So, let's see. What we do is we place a small ultrasound receiver on the tooltip, and because we know the precise timing of these acoustic beams, we use this receiver here to measure the signals that arise when acoustic beams impinge upon the tool. And because we know the precise timing, we can parse this time domain signal into a 3D matrix with its shape corresponding to the elevational and azimuth locations of the transmit beams. And if we do a simple search for the signal, for the maximum signal amplitude, we can find the position, we can find the rough estimate of the azimuth and elevation and range position of the tool. Now range is derived from just simple time of flight multiplied by the sound speed. And I said rough elevation and azimuth because the transmit beams in this sort of ultrasound imaging setup uses a sparse 2-degree beam spacing. So what we can do is we can take the actual signal magnitudes and interpolate a new finer max and to give us a finer estimate of the location. And methods for interpolation include both, they include cross-correlation, they include center of mass, but what we used is 2D Gaussian fit, and that's a picture of the Gaussian fit right there. So once we have the fine range, azimuth, and elevational coordinates, we can just convert these into a more convenient Cartesian X, Y, and Z framework. Now what's nice about all this is that the coordinates are all relative to the imaging probe. So what we have is perfect registration of the tool's position with respect to the ultrasound image. So this here is a photograph of the hardware setup that was required to implement everything that I just told you about. And in addition, we also have hardware here for the experiment to test for to validate for our accuracy. And I'll just take you through here really quickly. The centerpiece here is a Philips IE33 ultrasound scanner. It's attached to a 3D ultrasound probe. We use a ultrasound receiver as a mimic for the tracked tool. All these experiments take place within a water bath. This display indicates the user display that you see showing the ultrasound with the overlaid tracked tools. And then finally, we use an XYZ stage that's robotically controlled to provide ground truth coordinates against which we validate. This is what the user display looks like. Again, what you're seeing are two 2D orthogonal slices through a 3D ultrasound volume. And it's might be hard to see, but there's a blue line here. And that blue line corresponds to the location of the orthogonal slice. So this blue line indicates this slice, and this blue line indicates that slice. Furthermore, there's this sort of hot colored overlay. That is the overlay. That's the image enhancement we have to provide the tooltip. And on top of that, we have the XYZ coordinates displayed in real time. What's also nice about this method is that the slices are selected automatically based on where the tool is located, so you don't need to scroll through this large volume of 3D data to get the best view. So for the first experiment, what I had the robot do was translate the tool along these 4 rectangles, which represent the field of view for the 3D ultrasound imaging probe. And this red dot indicates the origin or where the actual probe is being held. And this movie that's sort of playing in the background is just an example of the display that we generated. Now, I let the robot run overnight, and this allowed us to generate close to 500,000 target registration error samples. And what these 2 plots show on both of their y-axes is TRE in millimeters. Now, for the plot on, I guess it would be your left, I'm showing TRE as it varies with the imaging depth. And in red is the mean TRE, and in blue is the standard deviation. On the right-hand side, this plot shows TRE as a function of the azimuth angle, or the angle of incidence from the tool to the ultrasound probe. And what this all tells us is that we have a mean error of about 0.36 millimeters. If we dig into the data a little bit more, we can plot a histogram of the X, Y, and Z biases, and this tells us that we have no significant biases in any direction throughout the imaging volume. And we can also plot relative distance error as a function of the distance between any 2 points we choose within our sample space. And what this tells us is if we have 2 sensors that are separated by greater than 3 centimeters or so, we'll have less than a 1% distance error. And what this suggests is that if we were to use 2 sensors to try to find tool orientation, we would have about a 1-degree rotation error. Really quickly for this next experiment, what I did was I took the tool and I rotated it about 180 degrees in 2-degree increments. And the purpose of this experiment was to test for the effect of the angle of incidence on accuracy. And as you can see here, mean error again was about 0.34 centimeters. So to conclude, I'd like to bring you full circle to the second movie I showed. Hopefully it has a little bit more meaning for you now, but what it means to us is that we were able to demonstrate the first real-time 3D tool tracking with image enhancement using ultrasound only. We have an accuracy of about 0.36 millimeters. We have the added benefit of perfect image registration. And all this is clinically significant because it has the potential to make procedures faster and safer. There's no disruption to the clinical workflow, and there's no additional costly hardware required. So thank you. I'm happy to take your questions, and if we can't get to them, come to my poster tomorrow.
So thank you very much. We have time for one